Hey everybody, this is a short little video on the comparison of the catalytic enzymes that are involved in the glycolytic and gluconeogenic pathways. My name is Justine Tabor and I am a chemistry student at Western Oregon University, and this presentation is for my biochemistry class. So first I just wanted to shortly review the glycolysis pathway. Glycolysis is a 10-step pathway that converts glucose into 2 molecules of pyruvate. The 10 steps are regulated by catalytic enzymes, which are labeled here with numbers 1 through 10. There are 3 irreversible reactions in glycolysis that are regulated by the enzymes hexokinase, phosphofructokinase 1, and pyruvate kinase. These are the first, third, and 10th steps of glycolysis. Here are the 3 irreversible reactions of glycolysis. You can see that these reactions all have large negative delta G values. Remember that reactions are spontaneous at negative free energy values, and these values are all largely negative, meaning that they are irreversible. Gluconeogenesis is the conversion of pyruvate to glucose, the opposite of what glycolysis does. In order to bypass the 3 irreversible reactions of glycolysis, the gluconeogenic pathway utilizes different enzymes. Hexokinase is bypassed by an enzyme called glucose 6-phosphatase. Phosphofructokinase 1 is bypassed by fructo 1,6-bisphosphatase, and pyruvate kinase is bypassed by 2 enzymes, pyruvate carboxylase and phosphoenolpyruvate carboxykinase, which is also known as PEPCK. The first bypass reaction of gluconeogenesis is catalyzed by 2 enzymes. The first enzyme is pyruvate carboxylase and catalyzes the conversion of pyruvate to oxaloacetate. The cofactor for this enzyme is biotin and fixes CO2. The binding of CO2 is activated by the reaction ATP converted to ADP and inorganic phosphate. This is then transferred to pyruvate to make oxaloacetate. This reaction takes place in the mitochondrial matrix. Since there are no channels to transport oxaloacetate out of the mitochondria, it is converted to malate. Once it is converted to malate, it can pass through the mitochondrial membrane through transporters into the cytosol, where it is converted back to oxaloacetate. Once in the cytosol, the second enzyme of this step is used. Phosphoenolpyruvate carboxykinase, or PEPCK, then converts oxaloacetate to phosphoenolpyruvate by using the reaction of GTP to GDP. The next bypass reaction is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. This reaction is catalyzed by the gluconeogenic enzyme fructose 1,6-bisphosphatase. The fructose 6-phosphate is formed by the hydrolysis of the phosphate ester at carbon 1 of the fructose 1,6-bisphosphate. Fructose 1,6-bisphosphatase is an allosteric enzyme. The last bypass reaction of gluconeogenesis converts glucose 6-phosphate into glucose. The enzyme glucose 6-phosphatase catalyzes this reaction by hydrolyzing glucose 6-phosphate. Note that the liver can send glucose to the blood to maintain homeostasis of blood glucose levels. This slide is just a short recap. Gluconeogenesis is not the reverse of glycolysis. We took a look at the 3 irreversible reactions of glycolysis and the 4 enzymes that are used in gluconeogenesis to bypass these reactions. It is important to note that the other 7 steps in both the glycolytic and gluconeogenic pathways share the same enzyme, no matter which direction molecules are being converted. And that's it. Thanks for listening, and I hope this video was helpful in understanding the differences in the catalytic enzymes used in glycolysis and gluconeogenesis.